Hello everyone, I'm Christy Risk, Senior Editor at GenomeWeb, and I'll be your moderator today. The title of today's session is Harnessing the Power of the Expi Protein Expression Systems in Vaccine Development. This e-case study will start with a brief presentation and will follow up with a Q&A discussion with our panelists. Our panelist today is Dr. Roland Leathers, Senior Manager of Strategic Alliances for the Europe, Middle East, Africa, and Asia-Pacific regions at Thermo Fisher Scientific. Dr. Leathers will be speaking about what makes the Expi systems unique and how they can be used in the development of vaccines. Special thanks to our sponsor, Thermo Fisher Scientific, for making this presentation possible. Dr. Leathers, please go ahead.
Christy, thank you very much indeed for the introduction. Hello, ladies and gentlemen, and thanks for joining us for this presentation. Since the beginning of this year, we've all been witness to the global coronavirus outbreak, which has now taken pandemic proportions. Researchers have quickly responded to the outbreak of this novel coronavirus, and using techniques such as next-gen sequencing and cryo-electron microscopy have already determined the virus's genomic sequence as well as its 3D structure. Armed with this information, considerable efforts are now being put into the development of recombinant vaccines to be able to protect global populations from disease and ultimately bring disease progression to a halt. At the same time, other groups are developing serological tests that will enable the detection of the presence of the anti-COVID-19 antibodies in the serum of patients to better understand the degree of patient exposure to infection and to make assessments of population immunity. Both recombinant vaccine production and serological test development have a requirement for high-quality recombinant coronavirus antigen. In this presentation, I would like to take a look at how we can harness the power of the Expi Protein Expression Systems to produce recombinant antigens for the development of vaccines and serological tests. So let me start with what is protein expression. Now, naturally, I'm sure that you're all very much aware of what protein expression is, but I think this serves as a good reminder that cells are factories. And in protein expression, we are simply exploiting a cell's ability to produce protein and retasking it to produce our desired protein of interest, which in this case will be the various coronavirus antigens that will have been genetically encoded into an expression construct. The efficiency of the protein expression system will ultimately determine both the yield and the quality of the expressed proteins. So if we put this into the vaccine researcher, what are the needs with regard to recombinant protein expression systems? Well, certainly the expression system must have the ability to produce vaccine candidates rapidly. Certainly during the exploratory phase of vaccine development, we need to be able to produce and evaluate numerous candidates. And then as we progress them, we would like to shorten the time to clinic. Equally, we need to be able to produce high yields of protein. Again, during the exploratory phases, we're after high yields because typically we're working with low culture volumes, and so we need to make functional assessments of the proteins being produced. Having generated our candidate vaccines, one then needs to be able to scale manufacturing quickly and cost-effectively. So here's an overview of the vaccine development workflow. You can see the exploratory phases, the developmental phases, and the manufacturing phases here. Over on the left-hand side, we see pathogen identification, followed by candidate vaccine panels, which will have been generated based on sequence information from the virus itself. Having screened panels of candidate vaccines and selected appropriate candidates for further development, frequently groups will then go on to make stable expression cell lines from those candidates. The stable cell lines will be further evaluated and eventually used in pilot scale manufacturing for potential clinical evaluation. Assuming that clinical evaluation is successful, after various rounds of evaluation, the candidate will be moved into large-scale manufacturing. So where do our Expi systems fit into this workflow? Well, certainly our Expi transient expression systems fit very nicely into the screening of candidate vaccine panels, but you will also see later in this presentation that we now offer you a path with the Expi systems through to, from transient expression through to stable cell line generation. makes these Expi systems unique? These Expi systems are complete systems. They are fully optimized systems. Each component in the system has been optimized individually and both as a system using design of experimentation approaches. Each system comprises cells, and these cells have been selected for their high specific protein productivity. In all cases, they are grown as single cell suspensions at very high cell densities. The media systems are all chemically defined, animal origin free, and manufactured under cGMP principles. Each system has its own unique transfection reagent. We need to achieve high transfection efficiency at high cell density, and so we've had to develop new transfection reagents for that purpose. The systems are also provided with expression enhancers and feeds to further boost expression. And of course, we can also provide expression vectors, of which we have a whole family of vectors that can be used for expression in the Expi systems. Interestingly, each Expi system is also provided with what we call a technical control, so that you can ensure that the systems are functioning optimally in your laboratory prior to actually introducing vaccine candidates into the expression system. That, I would now like to take a look at some of the Expi systems that are available to you. And the first system I would like to describe is the so-called Expi293 expression system, which is a mammalian cell-based expression system that uses a HEC293 cell line. Now, this system enables the rapid development of vaccine candidates and generates high yields of protein. Typically in this system, we have seen up to 1 gram per liter of protein being produced in transient transfection. And again, this is an integrated system, meaning that all of the components of the system have been individually optimized, as well as the system itself. The components of the system are the Expi293F cells, which have been adapted to high-density suspension culture, the chemically defined expression medium, which is transfection compatible, as well as the unique transfection reagent used to transfect your constructs of interest into the high-density cell lines, cell cultures. The second I would like to describe to you briefly is the ExpiCHO expression system. This is also a mammalian expression system that uses a Cho-S cell background for expression. This system provides a high-yield, end-to-end platform enabling a faster time to clinic. When used in transient mode, we see yields of protein up to 3 grams per liter in this system. It's also importantly that this system provides you with just one cell line that you can use all the way from discovery through to the clinic. This means that your exploratory work can be done using our research use only ExpiCHO-S cell line, exploring vaccine candidates, but as you progress them through to development and eventually through to manufacturing, you can work with our cGMP banked cell line, either in transient mode or to produce stable cell lines for more clinically relevant work. And of course, this is very important, being able to use the same cell line throughout the development process, because it enables you to maintain protein quality and function throughout the entire workflow process. The ExpiCHO system, similar to the 293F system, comprises the cells, which have been again adapted to high-density cell suspension culture, the chemically defined animal origin-free expression medium, the novel transfection reagent that allows transfection of these high-density cell cultures, a technical control. This is the antibody expressing positive control, again enabling optimization of the system in your lab prior to actually expressing your own constructs, the OptiPro serum-free medium for complexing the DNA vectors that will be transfected into the cell background, as well as ExpiCHO stable production media, which is a media formulation that's been developed specifically should you decide to take your constructs into stable cell lines. So I'd really like to reinforce that there is an easy transition from performing transient work through to performing stable work with the ExpiCHO system. On the left-hand side, you see the complement of ExpiCHO transient tools, which are research use only. But should you decide to progress candidates towards the clinic, you can switch to our cGMP-banked ExpiCHO-S cells, along with our stable production medium, which provides you a very nice transition from exploratory work through to development and again through to the clinic. And again, this is really the main benefit here, is that you can maintain the quality and the function of your protein from early exploratory work through to development and manufacturing. So in the next slide, I've simply really overlaid our vaccine development workflow diagram that you saw previously to show you where the ExpiCHO systems will be used. So typically we're seeing that the ExpiCHO transient systems are being used in the screening of antigens. These are the vaccine candidates. Once candidates have been selected for downstream progression, then groups will frequently switch to our ExpiCHO stable platform, enabling stable cell line generation in a cGMP-banked cell line, clinical-grade manufacturing of pilot batches of vaccine for evaluation, and eventually through to large-scale manufacturing. The third I would like to describe is the Xbsf expression system. This is an insect-based expression system that uses the SF9 cell background, and it provides a cost-effective solution for vaccine discovery. Interestingly, in head-to-head studies, we see that this system outperforms all existing insect platforms with at least 3-fold higher yields of protein. This is also the first insect expression system that uses a chemically defined medium. There are no yeast-related present in this medium. And we also provide the system with expression enhancers to further boost expression. By working with chemically defined media, of course, we can ensure that we have consistent cell growth and consistent protein expression yields run after run. The ability to actually make baculovirus in cell suspension cultures means that we have very short times from construct to protein. So there is no requirement for viral amplification using the Xbsf system, which means that we are already generating protein from constructs within 6 to 10 days, which is approximately half that of most other expression systems, insect expression systems. The components of this system comprise, again, the Xbsf9 cells, which have been adapted to high-density cell suspension culture, the chemically defined media, an enhancer, a protein expression enhancer, a transfection system for transfecting backmids into the SF9 cells, the back-to-back expression system, the DH10 back competent cells for generating the recombinant backmids, as well as the optimem-reduced serum medium used for complexing DNA prior to transfection. The final part of this presentation, I would just like to introduce you briefly to 2 case studies. The first here describes the work of Dr. Keith Chappell at the University of Queensland in Australia. Dr. Chappell is part of the Coalition for Epidemic Preparedness Innovations, otherwise known as CEPI. And in their work, following acquisition of viral sequence information from patient samples, the group have gone on to design expression constructs using the University of Queensland's so-called molecular clamping technology, which maintains the natural 3D structure of the expressed antigen. Throughout the work that Dr. Chappell has been performing, the ExpiCHO system has been used as the system of choice because it provides fast, high yields, and quality protein. If you are interested in understanding more about the use of the Expi system in Dr. Chappell's work, I'd like to refer you to the recent publication that's cited at the bottom of this slide. In the second case study, I'll be describing briefly the work of Dr. Jiang Zhu at the Scripps Institute in La Jolla, California, whose team are developing vaccines against HIV this time. During the course of Dr. Zhu's work, they've used extensively the ExpiCHO system to express native-like trimers and G140 nanoparticles. And they found that the ExpiCHO system allowed them to improve both the yield and the quality of the GP140 nanoparticles. If you're interested in understanding more about the use of the ExpiCHO system in Dr. Zhu's work, again, I'd like to refer you to the citation at the bottom of this slide. Summary. I've introduced you to 3 Expi Expression Systems. Firstly, the Expi293 transient expression system, which uses a HEC293 suspension cell line to provide high yields of protein up to 1 gram per liter. Secondly, the ExpiCHO system, which uses a Cho suspension line for transient expression work with yields of up to 3 grams per liter. But the system also provides a path through to the clinic through the provision of a cGMP-banked cell line that can be used to generate stables for development and biomanufacturing work. Finally, the Xbsf system for generating high yields of protein from an insect cell line maintained in a chemically defined, yeastolate-free medium for reproducible expression performance. If you'd like to know more about any of these systems, then please refer to the web link below or contact your local Thermo Fisher representative. Thank you very much.
Thank you, Roland. Let's start the Q&A. So how do the protein yields from the ExpiCHO expression system compare with from Freestyle Cho or the Expi293 system?
Thank you, Christy. Well, working with various test proteins from different protein classes, the ExpiCHO expression system was found to yield between 25 to 160-fold more protein than the Freestyle Cho expression system, and we see between 2 to 4-fold more protein than the Expi293 expression system.
And do I need to change media after transfection in the Expi293 and ExpiCHO systems?
No, you don't. In fact, that was one of the design prerequisites of these projects. So the media systems in both the Expi293 and the ExpiCHO system have been designed to be transfection compatible, and there is no requirement to change the media post-transfection.
Can the Expi systems be used to express genes that could then be used in the clinic?
Well, the culture media and the transfection reagents used in the Expi293 and ExpiCHO systems, and in fact as well as the Xbsf system, are all chemically defined, animal origin free, and are all manufactured under cGMP. Each of these systems is provided with a cell line for research use only. However, we also have cGMP-banked master banks for the Expi293 and the ExpiCHO cell lines. So for those groups seeking to express protein for downstream use in the clinic, we are able to provide a complete and fully cGMP-compliant expression system, along with the documentation that will be required by the regulator.
And how cost-effective are the Expi systems for expressing proteins of interest?
Well, all 3 Expi systems, the Expi293, ExpiCHO, and Xbsf, represent the highest yielding expression systems available in the market today and would normally be expected to provide you with the lowest cost milligram of protein that you have probably ever produced.
How many days of culture required before harvesting protein with the Xbsf system?
Well, depending on the particular protein being expressed, the harvest time will typically be between day 6 and day 10 post-transfection. Harvest time is typically a parameter that we would recommend should be included as part of your expression optimization process, as it can impact protein quality. We typically find that the whole process of generating baculoviral stocks through to harvesting protein is roughly half that of other baculoviral systems available today, and this is because we're able to eliminate the need for viral amplification steps prior to initiating our expression runs, thereby significantly reducing the time it takes to generate protein from novel constructs.
And where can I find additional information on the Expi systems?
Well, for additional information, I can refer you to thermofisher.com/protein expression in one word. Otherwise, please take contact with your local Thermo Fisher representative.
Great, thank you very much. We'll wrap it up there. We'd like to thank panelist Roland Leathers and our sponsor, Thermo Fisher Scientific. Thank you all for joining us for this session.